In today's lecture, we're going to look at the circulatory system. Within the circulatory system, we're going to look at the blood, we're going to look at the heart, the blood vessels, the immune system, and the lymphatic system. So this is a really big system to go through at one shot. So you may want to break this video up into a couple of different times that you go through things the first time you're going through it, just to make it a little bit easier to understand. So functions of our blood. It's going to transport things, nutrients, waste, oxygen. It's going to help in regulation of temperature and pH, and then it will provide protection through immunity and clots. Over here, there are a couple of illustrations of some red blood cells. You can see them also in the picture down here with our red blood cells, that they're a biconcave disc. So the cell is actually flattened out in the middle, and you can see this flattening here on these cells. This is an example of a crenated red blood cell that is dried out, and here is one that is swollen. So when we look at our blood, it's 38 degrees Celsius or 100.4 degrees Fahrenheit. We think of normal body temperature as being 98.6. That's measuring it on a mucous membrane. This would be measuring it like you would with a meat thermometer. That's the reason we don't do this temperature normally on a person, is nobody really wants to have a thermometer stuck in them like when you're cooking a turkey. But it is a little bit warmer inside. pH is 7.35 to 7.45. A female will typically have 4 to 5 liters of blood, a male typically 5 to 6 liters. It is dependent on size, so if you're a 6'2" female, you're probably going to be above that 4 to 5 liters. Likewise, if you're a 5'2" male, your volume's probably going to be on the lower end of things. And it's going to be made up of the plasma and the formed elements. So when we look at the blood, it would not be very efficient to count all of the blood cells. There's way too many of them. And if we were to take them out, the person's not going to survive without their blood. So what we do is we look at this, we take a small sample and we do the hematocrit, which looks at the percentage of the total blood volume that's occupied by the red blood cells. What is the concentration of the red blood cells in what we take out? The plasma is the liquid portion of the blood. It's 91.5% water, 7% protein, and 1.5% other things. This makes up 55% of the total blood. The albumins are 54% of that protein. They're made by the liver and they help to control osmotic pressure. The globulins are 38%. They will consist of the antibodies and immunoglobulins, which are your immune proteins. The fibrinogen is 7% of the protein. That's involved with the formation of clots. When we looked at the formed elements, they're 45% of the total blood. The red blood cells are going to be the ones carrying oxygen. You'll have between 4.8 and 5.4 million of those. And then the white blood cells are immune cells. This is a picture here of a blood smear. This would not be a typical blood smear you would see in a person, but it's a great example to use for teaching purposes because it's got everything on one slide. These here are your red blood cells. These are your PMNs or neutrophils. So they're called PMN because of being a polymorphonucleoside. They'll look like the nuclei have multiple different shapes in there. This is also a PMN. Here is an eosinophil. It stains red. A basophil has this blue staining. And our lymphocytes here. And then a monocyte will have this bigger C-shaped nucleus. So when we look at the white blood cells, they're also called leukocytes. Leuko means white, cyte is a cell. Red blood cells are erythrocytes because erythro means red. So you're going to have between 5 and 10,000 of these. Some of them are granulocytes because they have granules in them and a granular appearance. This includes the neutrophils, eosinophils, and basophils. The neutrophils, also known as PMNs or polymorphonucleosides, are 60 to 70% of the white blood cells. The eosinophils are 2 to 4%. The basophils are 0.5 to 1%. The agranulocytes do not have the granules in them. This includes the B and T lymphocytes. T lymphocytes have a couple of main subsets. the TC or cytotoxic T cells, and the TH or helper T cells. With the B lymphocytes, we have memory B cells and plasma cells. Their job is to secrete antibody. Our NK cells, it stands for natural killer cells, they're a non-specific immune cell, so they're not going to be as picky about what they kill. The monocytes are 3 to 8%. The platelets, these are fragments of a giant cell called a megakaryocyte. It will break down into membrane fragments, and you'll have 150,000 to 400,000 of those. So when we look at blood cell formation, we call this process hemopoiesis or hematopoiesis, the process of making the formed elements. A lot of it is going to take place in the red marrow, which is your vascularized connective tissue in the spaces between the trabeculae of the spongy bone. This diagram here shows you starting with this pluripotent stem cell. Pluripotent stem cells have the ability to form several different cell types. So on here you can see this one moving into lymphocytes, the megakaryocyte, red blood cells, monocytes, neutrophils, basophils, eosinophils. These things like the IL-7 or EPO are chemical signals that are going to tell it which type of cell to develop into. We divide these into two main pathways. All of these are the myeloid pathway, and they will become a myeloid stem cell in the red marrow. Those can form your red blood cells, platelets, eosinophils, basophils, PMNs, or monocytes. The other option is to become a lymphoid stem cell. Coming over here, they undergo incomplete development in the red marrow and go to the lymphoid tissue to make your B and T lymphocytes. The vast majority of them are myeloid stem cells. If you get sick and you need some specific immunity, you can have some of those shift over to become lymphoid stem cells when they're needed. So your red blood cell or erythroid structure is a biconcave disc. They're a small cell. They have no nucleus or organelles, but they do have hemoglobin, which is the pigment that allows them to carry oxygen. So our red blood cells are going to live for 120 days. So every 120 days, in theory, you should have all of your red blood cells recycled. They don't all die on day 120, and then the next day you get all new ones. You're going to lose some every day and make some new ones every day. So your old red blood cells are going to be broken down by macrophages in the spleen, liver, and the red marrow. So they'll be phagocytized, and then they will be split into the heme portion and the globin portion. The globin portion will be broken down into the individual amino acids that make it up, and they can be reused. So they go into the pile of amino acids you have as your building blocks. The iron is going to be removed from the heme, and it will attach to transferrin. So ferrous means iron, so this literally means transfers iron. This will go to the red marrow, and it will be used for new hemoglobin synthesis. So the body really does try to recycle a lot of its iron. So then it can do erythropoiesis again in the red marrow. So then you can have, after the iron's removed from the heme, you'll have the non-iron portion of the heme. Some of it will be converted into biliverdin, then bilirubin, and bile that you'll use in the gastrointestinal tract. Some of the bilirubin can be converted into urobilinogen in the large intestine, and then some of the urobilinogen in the blood and urobilin to make yellow pigment in the urine. And then most of the urobilinogen will end up in the feces as a brown pigment called stercobilin. So with the red blood cell recycling in there, the reason females do need a slightly higher amount of iron intake when they're of childbearing age is because the ones from the red blood cells that are recycled, you're going to recycle that iron. the blood that's lost to the menstrual cycle, that iron is not recovered. So when we look at things that influence red blood cell production, one of the things is hypoxia. It's a deficiency of oxygen. If this goes on for a prolonged amount of time, the person can develop cyanosis, where they'll have bluing of the fingertips, the lips, any of the tissues where the blood vessels are really superficial and close to the skin. You can see the blood turning blue. Erythropoietin, or EPO, is the hormone that's made by the kidneys. Since they do all the filtering of the blood, they would notice that we need more red blood cells. That will stimulate the red marrow to do erythropoiesis. So we do have synthetic forms of this that can be used particularly for cancer patients, and they can be really helpful to give them more red blood cells because some of those can be killed off by their chemotherapy regimen. So getting the blood counts up a little bit can actually be really helpful to give them a little bit more aggressive treatment protocol or have them feel better throughout the process. So looking at white blood cell functions, neutrophils like to do phagocytosis. They've got lysozyme. Their snack of choice is bacteria. However, neutrophil levels can be elevated for numerous other things. So if you're looking on a blood count and you see neutrophils elevated, one of the possibilities is bacterial infection, but there are other things that can cause them to be elevated. Eosinophils work along with histamine. Histamine is the stuff that you would have during allergies that's going to cause redness, swelling, runny nose, itchy eyes, a lot of those unpleasant things in there. So you will see these being elevated in allergies as well as with parasitic worm infections. Basophils are going to release histamine and other chemicals for inflammation, so they're also involved with allergic reactions. Our B cells, these are going to include plasma cells and memory B cells. Their job is antibody formation. With the T cells, they're going to help promote immune function or be cytotoxic. So your TC are your cytotoxic T cells. They kill an entire infected cell. If something's infected with a virus, there's no way of getting it out of the cell. The only option for the body is to kill that infected cell. The T helper cells, we do have more than just two types of these, but these are the two primary populations. The Th1 are going to promote an infection type response, while the Th2 are going to promote more of your allergic or hypersensitivity type responses. The NK cells, or natural killer cells, these will target various things, but it does include cancer cells and microbes. The MHC is your major histocompatibility complex. These are proteins for identifying self cells. These are really important because this is a way for your immune system to be able to identify cells that are yours and leave them alone. So platelets, these are going to form the plug in clotting. They'll release chemicals for vasospasm and to promote clotting. So when there is a hemorrhage, one of the first things that will happen is a vascular spasm to try and reduce the amount of blood loss to that area. Then you'll have platelet adhesion. So they're going to start to stick to the area around that. A platelet release reaction is going to call additional platelets into the area, and they will begin to aggregate. They'll stick to each other. Then you will form fibrin that will help to form fibers across in helping to hold platelets into place. So this process is called coagulation or clotting. When we look at human blood groups, these are expressed codominantly. What that means is you'll inherit a trait from mom and one from dad, and you're going to put both of them on your blood cells. So if a person is blood type A, they would either inherit A from both parents or A from one and O from the other. A makes the A marker, B makes the B marker, O does not make any marker. So that's why inheriting A from one and O from the other makes the person type A. They're going to make antibodies towards the markers they don't have. So they will make anti-B antibodies. For a person who's type B, they'll make anti-A antibodies. They can inherit B from both parents or B from one and O from the other. A person who is AB inherited A from one parent and B from the other. they will not make antibodies to either one because they have both on their cells. The person who is type O, they're going to make antibodies towards both, but they inherited O from both parents. So that's one of the markers on the red blood cell. The other one that we commonly look at is the Rh group. So when we talk about being type O positive or O negative, this is the positive or negative that we're talking about. So the Rh group is called that because it was first discovered on a rhesus monkey. Rh positive is the dominant trait, and Rh negative is the recessive trait. So if you have one parent that is Rh positive and one that is Rh negative, the child will be Rh positive. So if you have an Rh negative mother and she's pregnant with an Rh positive child, her body will develop antibody towards that. So we give an injection of RhoGAM to prevent that from happening. So now looking at the heart, with this illustration of the heart, if you printed your notes out in black and white from my class, you may want to highlight this red line on here that we'll refer back to later when we talk about the electrical conduction of the heart. So the base is the top side of the heart. So this would be the base running along this line. The apex is at the bottom. It's a pointy part. It's going to be on the left side. So the ventricles are the two lower chambers. Blood is going to exit out these. The atria are the two upper chambers where blood will come in. The heart is going to have a series of membranes covering it. The pericardium is going to be the membrane that surrounds the heart cavity. It helps to hold the heart in place. The outer portion of it is fibrous, so it's a tougher, more protective surface, where the inside is the serous pericardium. It's thinner and more delicate, so it's similar to what we had with the articular capsule in the joint. With organs, we talk about parietal and visceral layers. A parietal layer is going to form the sac around the organ, so in the case of the heart, the pericardium is a parietal layer. The visceral layer is going to be the inner membrane that would adhere directly to the organ. So in the case of the heart, that will be the epicardium. It will be adhered directly to the heart. The pericardial fluid is going to be between the pericardium and epicardium. It's going to help to reduce friction in there as the heart moves. So that pericardial cavity has that pericardial fluid in it. The mediastinum is the cavity that holds the heart. This is the space between the lungs. So if we look at the heart wall, going from more superficial to deeper, you have the epicardium that's the outer membrane. Myocardium is the muscle tissue. So it is cardiac muscle. So you're going to have intercalated discs, which are thickenings in the sarcolemma. And within those intercalated discs, you have little gap junctions that will allow the rapid conduction of action potentials. This is going to allow the heart muscles to contract together. Endocardium is going to be simple squamous cells that are going to line the inside of the chambers. It will fold back on itself to make the valves and tendons in the heart, and it will be continuous with the endothelium that will line the blood vessels. So the heart has 4 chambers. The atria are the 2 upper entry chambers, while the ventricles are the lower exit chambers. So you have 2 atria, 2 ventricle. The inner atrial septum is going to divide the atria into right and left areas. The interventricular septum is going to divide the ventricles into right and left areas. The foramen ovale is an opening in the fetal heart that's supposed to be there, and it will close after birth. In a fetal heart, it's not going to be getting oxygen from the lungs. It's going to get oxygen from the umbilical cord. So instead of sending blood to the lungs to be oxygenated, the foramen ovale will allow you to bypass the lungs. So when we look at major vessels in circulation, I've tried to color code these notes a little bit here. When we talk about arteries, that's a vessel that's moving away from the heart. A vein is a vessel that's returning to the heart. Oxygenated blood is red. Deoxygenated blood is blue. Not all arteries are red, and not all veins are blue. So your superior and inferior vena cava. These are going to drain deoxygenated blood from the body, and it's going to come back into the right atria of the heart. The coronary sinus is going to drain deoxygenated blood from the heart tissue. So the heart tissue is going to need to be fed as well. The pulmonary trunk is going to take deoxygenated blood from the heart to the lungs. So it will come into the right atria being deoxygenated, and it's going to come out the right ventricle through the pulmonary trunk. Being that you've got two lungs, it's going to split into the pulmonary arteries to go to the right and left lung. In the lungs, it will pick up oxygen. Then you're going to have oxygenated blood returned back to the heart through the pulmonary veins. It will come into the left atria, and then it will go out through the left ventricle, and then eventually lead out the ascending aorta that's going to carry that blood from the heart to the body and the heart muscle itself. So the coronary arteries will branch off to feed the heart itself. So valves are important in the heart because you want to prevent backflow of blood. If blood starts to flow backwards, you're going to potentially have mixing of oxygenated and deoxygenated blood, and the system's not going to be moving efficiently. So the AV valve, or the tricuspid valve, it's the right AV valve, would be between the right atria and the right ventricle. The left AV valve, also called the mitral valve or bicuspid valve, would be between the left atria and the left ventricle. The tricuspid is called that because of having 3 flaps. Bicuspid has 2 flaps. The semilunar valves, you'll have a pulmonary and an aortic. The pulmonary is between the right ventricle and the pulmonary artery. The aortic is between the left ventricle and the aorta. The chordae tendineae are little cords that are going to attach to the valves to prevent them from opening backwards in the ventricles. The papillary muscle is going to connect those chordae tendineae to projections on the inside of the cardiac muscle. If you look inside of a heart, it actually looks a lot like what it looks like in your mouth if you lift up your tongue, with little ridges, and the valves look a lot like the little flap of skin that's underneath your tongue. So if you're curious, you can usually get hearts from the butcher shop to cut open and look at. They are going to be from an animal, but they'll look very similar to a human heart. And if it's in the fall, Thanksgiving time, a lot of times your turkey's going to come with one. It'd be small, but you can still get an idea what the tissue's like. So the coronary circulation is going to be to the heart itself. You're going to have left and right coronary arteries that are going to carry oxygenated blood to the heart muscle. Those will branch off into additional arteries to try and spread that blood over the heart. The coronary sinus is going to carry the deoxygenated blood from the heart back to the inside of the heart to be recirculated. One of the things that can happen is the formation of anastomoses, where you will have branches of arteries that will start to connect. Basically, it's like creating little bypasses in the heart. So this will occur when there is more blood flow going to an area. This occurs as an adaptation to the heart in somebody who does a lot of cardiovascular exercise, as a way of feeding the heart muscle more efficiently. This is actually a very beneficial thing for the heart, because if one of those pathways gets plugged, there's an alternate route that the blood flow can take. When we look at the conduction of the heart, you can always refer back to that first picture that had the red lines on it if you'd like. The pacemaker is a special group of cells that's going to generate an action potential in a rhythmical pattern. Your main pacemaker is the SA node. This is where cardiac excitation is going to begin. This is the one that's going to speed up your heart rate when you start moving. It's located in the right atrial wall. The AV node is in the inner atrial septum. This is going to slow down the action potential. We want to do this because we want time for the atria to contract before the ventricles contract. If all 4 chambers contract at the same time, blood's going to squirt out in all 4 directions. That's not what we want. We want the blood to go from the atria into the ventricles, and then the ventricles to the lungs and the body. So the AV node is going to create that delay to allow the atria to empty into the ventricles. This will then spread down the AV bundle or bundle of His. It's in the interventricular septum, and it's going to conduct the action potential down from the atria to the ventricles. The right and left bundle branch is going to split and carry the impulse around the apex of the heart. And then the Purkinje fibers will spread out over the ventricles and rapidly conduct the action potential from the apex to the remainder. When we look at the cardiac cycle, the relaxation is going to be at the end of the cycle. While it's relaxed, the heart's going to repolarize. There's going to be a decrease in pressure, and it will allow the AV valves to open and the ventricles to passively fill by about 75%. Systole is the contraction phase, diastole is the relaxation phase. So when you have systole happen again, it will fill up that last 25% of the ventricles, and then the ventricles will contract. So the heart does make some sounds. You can feel them on your chest, and then they can be heard with a stethoscope. So lub is the first sound. It's sometimes referred to as S1. The sound is created by the AV valves closing. Dub is the second sound, or S2. It's created by the semilunar valves closing. So the ECG or EKG is the electrocardiogram. This is going to measure the electrical activity on the heart. And in here, we're just concerned about knowing the parts on a normal ECG. However, somebody that reads these all day long would be able to see a lot more information from an abnormal one than you would be able to just studying the normal ones here. So the first wave is your P wave here. This represents atrial depolarization. So this is looking at a heartbeat of, say, around 72 beats per minute to give you these relative times. Don't worry about memorizing them for my class. If you speed the heart up, all of these relative times are going to get shorter. If you slow it down, these relative times are all going to get proportionally longer. So the P wave, atrial depolarization, lasts about 0.1 second. The QRS complex, this is the onset of ventricular depolarization. This lasts about 0.3 seconds. This little delay here is what allows the ventricles to fill. The T wave here is ventricular repolarization. So the ventricles are repolarizing, lasts about 0.4 seconds. The atria do repolarize, it's just masked by the activity of the ventricle depolarizing here. So we have a couple of equations that will explain behavior of the blood and the blood pressure. The first of them being cardiac output. This is how much blood is ejected from the left ventricle of the heart in 1 minute. We look at the left ventricle because that's what goes to the body. 1 minute because we need to have a time frame to work with. So your stroke volume is how much comes out of the left ventricle in one contraction. Your heart rate is how many times it contracts per minute. So our equation is CO equals SV times HR. Cardiac output is stroke volume times heart rate. The reason we care about cardiac output is blood pressure is your cardiac output times the total peripheral resistance. The total peripheral resistance is the resistance of the vessels to having blood flow through them. So we can look at each of these components individually. So things that would control stroke volume, one of them is Starling's law. Starling was a researcher, has a couple of laws, he named them after himself. So Starling's law of the heart says the more the heart stretches, the more it has to contract. So this will control the stroke volume. The other thing that influences this is the forcefulness of contraction. This is controlled by the autonomic system. So the harder the heart contracts, the more volume is going to come out of it. So this will be influenced by calcium levels, digitalis, which is a substance derived from the foxglove plant, anoxia, which is an absence of oxygen, acidosis, anesthesia, and potassium levels. The next thing is the pressure to eject blood. So it's not unusual for a little bit of blood to remain in the ventricle after contraction. If you imagine pouring out a glass of water, there's a little bit of water left after you pour it out. Not a big deal. The problem is if it continues to do this and you start to accumulate more and more being left in the heart. Then it starts to back up, and eventually it's going to exert force on the pulmonary trunk. So this is what would happen with congestive heart failure, is the blood starts to back up into those other tissues, and then those blood vessels are going to start to ooze out some of the fluid from there, and you're going to have fluid in the lungs and edema or fluid in the tissues. So congestive heart failure can progress rapidly, but it can be treated with diuretics. So a person can actually lose a lot of fluid from those diuretics if it's allowed to get out of hand. So then looking at things that affect heart rate, it's under autonomic control. So we know sympathetics are going to speed it up. They're inhibited by anoxia, acidosis, anesthesia, and high potassium levels. The sympathetics are increased by epinephrine and norepinephrine, higher calcium levels, and digitalis. The cardiac accelerator nerve is going to be the one that delivers the message to increase the heart rate, and it's going to use norepinephrine. It's going to get that information from the cardiovascular center. The cardiovascular center is in the medulla oblongata. It's going to take input from sensory receptors, higher brain centers like the limbic system and the cerebrum, and decide to either increase or decrease the impulse with a message through the sympathetics or parasympathetics. If it's going to send a parasympathetic message, it'll go through the vagus nerve or cranial nerve 10. It will take information from the cardiovascular center to deliver a parasympathetic message, and it will decrease SA node activity using acetylcholine. So we have baroreceptors that will sense pressure changes. These are located along the aorta and the carotid artery. So with the carotid artery, it wants to make sure there's adequate pressure to the head, to the aorta, making sure there's adequate blood flow coming out of the heart. We also have chemoreceptors that will sense chemical changes, such as changes in levels of oxygen, carbon dioxide, and the hydrogen ion. Other hormones can influence this. Epinephrine and norepinephrine will increase the heart rate and contraction force. Thyroid hormone can increase the heart rate as well. When it's too fast, we refer to it as tachycardia. When it's too slow, we refer to it as bradycardia. Exercise, stress, and excitement all stimulate the adrenal medulla and speed up the heart rate. So ions can influence this. The body will monitor levels of potassium, sodium, and calcium. Other influences can be your age, your gender, fitness levels, and body temperature. So looking at the heart and exercise, aerobic versus anaerobic exercise. While there are benefits to anaerobic exercise, the heart really likes aerobic exercise. Over time, it will adapt and increase the cardiac output and metabolic rate. The heart will respond to increased demands. So if you stop the exercise, the heart is not going to keep those adaptations. It will view them as no longer being necessary. So sometimes people will look at it and they'll go, well, I was very active in high school. Well, if that was 30 years ago, the heart has no longer kept those adaptations. So looking at the blood vessels going to the body, arteries are considered the larger vessels. They will branch out into the arterioles, which are the medium-sized vessels. Those will branch out into the capillaries, which are the smallest vessels. These are going to allow for oxygen to go into the tissue and carbon dioxide to go back into the bloodstream. I've tried to color code these here to go along with circulation to the body, not the lungs. The venules are medium-sized vessels, and then those will come together to make the veins, which are the larger vessels. In this illustration here, you can see the artery on the bottom and a vein on the top. There is a significant difference in the walls of the arteries and the veins. There's a lot more muscle in an artery wall. When you're looking at them in a cadaver, they're going to look the same. You have to feel them to determine if it's an artery or a vein. So arteries are more flimsy, or arteries are more muscular, veins that feel more flimsy. They also look just like nerves, which are going to feel more rubbery. So the lumen is the hollow space inside of a vessel. So the space you'd have here in the middle. When we look at the structure, the endothelium is going to be the inner layer. It's made up of simple squamous tissue inside the basement membrane. And then the middle layer is myothelium or muscle. And the outer portion is elastic tissue and collagen. When we talk about vasoconstriction, in general, that's narrowing the lumen, and it's a sympathetic response. Vasodilation is going to increase the size of the lumen, and in general, that's a parasympathetic response. So some of our arteries are elastic in nature and some are more muscular in nature. Your elastic arteries have more elastic fibers in the middle layer. These are the larger diameter fibers. These need to be able to adapt to additional blood flow going through them. Muscular arteries are your medium and small S arteries. They have more muscle in the middle layers. This will allow them to be able to have greater flexibility in vasodilation or vasoconstriction. These are the ones that are going to adjust the blood flow. And then your capillaries, they are just simple squamous tissue, and it allows for the rapid passage of the gases across the membrane. So capillary exchange is when you move that oxygen into the tissue from the bloodstream and the carbon dioxide into the blood from the tissues. So this is largely controlled by diffusion. However, when we talk about the respiratory system, we'll look at other factors in there that will help to influence moving the carbon dioxide and oxygen. So in the veins, they don't have much muscle in the wall, so instead they're going to rely on having valves to prevent backflow. This is continuous with the endothelium. So to get venous return, there's very little muscle in the vessels. It's going to have to rely on contraction of the heart and other mechanisms we refer to as the pedal pump and respiratory pump. So the pedal pump is also known as the skeletal muscle pump. What happens is as skeletal muscle contracts, it's going to put pressure on the veins in that area to squeeze the blood through. So we call it the pedal pump because this area was first described looking at the calf muscles as a person would walk, squeezing those vessels in there and pushing the blood up through. So if a person does a lot of standing, but they don't move around a lot, it will allow the blood to pool in that area and can lead to more varicosities. Hemorrhoids are actually just a varicosity in the rectum. So if a person goes in and they just sit on the toilet and hang out in there, it's going to allow blood to pool in that area. If they go in there to take care of business, you're going to have muscle contraction that's going to keep the blood from pooling. So if you want to read, go sit somewhere else on a seat that's more accustomed to not having blood pool in that area. So the respiratory pump is going to work similar in the torso. As the lungs expand, it's going to put pressure on the veins to squeeze the blood through. We generally don't have problems with getting varicosities here. If you stop breathing, you're not going to be around long enough to worry about the varicosities. So another component of blood pressure is your total peripheral resistance. So your vascular resistance is the opposition of the vessels to having blood flow through them. So this may seem a little bit odd when that's the purpose of having the vessels, but anytime you have something flowing over another surface, you will usually have some resistance to flow. So there's three things that are going to influence this. The size of the lumen is one of them. If you decrease the size, it's going to increase the pressure. So you can see this effect if you put a nozzle on a garden hose or try to drink through one of the coffee stirring straws. Blood viscosity will also influence this. It's the resistance to flow. So if you imagine what it's like to try and drink honey through a straw versus water, honey is a more viscous fluid. And then vessel length. The longer the vessels are, the more pressure it will take to move the fluid. This is where we can really have an influence on our blood pressure. For every pound of body fat, you have between a half a mile and a mile of blood vessels in there. So eliminating that can actually really decrease some of the work for the heart. So when we look at blood pressure regulation, there's several components that come into play. Your cardiovascular center in the medulla oblongata is going to regulate the heart rate and the stroke volume. This is going to use both neural and hormonal negative feedback loops. The higher brain will have influence from the cerebral cortex, the limbic system, and the hypothalamus. You can actually have the heart rate increase in anticipation of it needing to go up. Mechanoreceptors, they're going to monitor joint movement. You'll see a rapid increase in heart rate at the beginning of exercise. Baroreceptors will sense pressure change. These may operate a little bit slow. Orthopneal hypotension is the name given to it when it operates too slow. This can happen when a person goes from either lying down to standing or even sitting to standing too quickly, and the pressure doesn't get up high enough to their brain, and they can actually lose their vision or lose consciousness and faint temporarily. Chemoreceptors, these are going to monitor levels of oxygen, carbon dioxide, and the hydrogen ion. It's going to help to tie this in with respiration. So you'll notice usually your respiratory rate will change when your heart rate changes as well. We have carotid bodies and aortic bodies. These are chemoreceptors in the carotid artery and the aorta. Again, making sure that you've got adequate supply to the brain and coming out of the heart. Your vasomotor tone, this is a moderate state of vasoconstriction. If you have a moderate state of vasoconstriction, it allows you to go in either direction and make adjustments to your blood pressure. So hormonal regulation. Understanding hormonal regulation is really important because a lot of how we pharmaceutically treat hypertension is by modifying hormone levels. So the renin-angiotensin-aldosterone system, this will work when there's a perceived decrease in blood flow to the kidneys. The kidneys will secrete renin into the blood, which is going to combine with the angiotensin-converting enzyme. This activates the hormone to become angiotensin 1 and then angiotensin 2. This increases vasoconstriction and increases blood pressure. It will also trigger the secretion of aldosterone that will lead to an increase in sodium reabsorption by the kidneys. This in turn increases the total blood volume and the blood pressure. With epinephrine and norepinephrine, when there's an increase in sympathetic stimulation, the adrenal medulla is going to increase the epinephrine and norepinephrine. This is going to lead to an increase in cardiac output by increasing the heart rate and contraction force. It will also lead to vasoconstriction of the vessels to the abdominal organs, increasing the blood pressure. ADH, antidiuretic hormone, or vasopressin. When there's a decrease in blood volume, ADH is going to be secreted by the hypothalamus and released through the posterior pituitary. This will lead to vasoconstriction, to increase blood pressure. It's also going to tell the kidneys to conserve water to increase the blood pressure. So all of these above are all going to increase blood pressure. Atrial natriuretic peptide is going to be the only one that decreases blood pressure. This is released by the atria to decrease the blood pressure by vasodilation and promoting the loss of sodium and water in the urine. This will decrease the blood volume and therefore the blood pressure. So in here I've put some categories of blood pressure medications. This is not all of them by a long shot. Different sources will divide these up differently. It's just to kind of give you an idea of some ways that things can be controlled. So one of them is the diuretics. What these will do is cause you to excrete more water. When you excrete more water, it's going to lower the blood volume and the blood pressure. So some of them may also cause vessels to dilate. The beta blockers or beta adrenergic receptor blockers, these block the sympathetic stimulation. So what they will do is they block the effects of the catecholamines so the heart does not beat as hard and widens the blood vessels. Calcium channel blockers, they can bind to the surface of the heart and vascular cells to interfere with the calcium flow so the heart won't contract as hard and widens the blood vessels. There are some that will act selectively on the blood vessels and not the heart. The ACE inhibitors and angiotensin II receptor blockers. ACE inhibitors are going to reduce the production of angiotensin, so the arteries are not able to constrict. This will allow arteries and veins to dilate and reduce blood pressure. The angiotensin II receptor blockers are going to bind angiotensin to these receptors and block its action on the heart, kidney, and blood vessels. And then we have other vasodilators that can expand blood vessels as well. So the pulse is going to refer to the number of contractions of the heart per minute. We refer to it as beats per minute. When we talk about systolic and diastolic pressure, the systolic pressure is the pressure during contraction of the heart, while the diastolic pressure is while the heart is relaxing and the blood is just coasting. So with the general circulation, we can summarize it here and follow it through this pathway. The blood from the right ventricle is going to go to the pulmonary trunk and arteries. This will lead to the lungs. It will oxygenate it. It will return through the pulmonary vein into the left atria, then down to the left ventricle, and out the aorta into the systemic circulation, delivered to the body, and then will return through the superior and inferior vena cava to come through the right atria and the right ventricle and repeat the whole cycle again. So with the hepatic portal circulation, normally the blood is going to go to one location and then return back to the heart. With the hepatic portal circulation, is going to make a detour or a second stop. It's going to go from the intestines, where it's going to absorb the nutrients, to the liver so that it can process the fats, toxins, and nutrients, and then it will return to the heart with the remaining circulation to be spread throughout the body. In the fetal circulation, we bypass the lungs in a fetus. So the blood is going to go from the right atria to the right ventricle, and instead of going from the right ventricle to the lungs, it'll go from the right ventricle to the left ventricle through the foramen ovale. Oxygenation will happen through the umbilical cord. Atherosclerosis is hardening and plaquing of the arteries. This is the result of fatty plaque and buildup in the vessels. Arteriosclerosis is due to calcification. That's usually secondary to something else going on in the body. So in this country, atherosclerosis is incredibly common. We actually see signs of it a lot of times in a 10-year-old child. So as adults, to think that you don't have any atherosclerosis going on is pretty much being in denial. And so we think a lot of it is due to diet and lifestyle with activity levels. So your lymphatics, these are going to drain excess interstitial fluid. It's an open system. With the blood, it's a closed system. You have a vessel that's going to take the blood there, a vessel that's going to bring it back. This is considered an open system because all it does is drain. The lymphatics are also going to transport dietary lipids. So your large fats will be deposited into the blood again by the heart, but they will go from the digestive system through the lacteals into the lymphatics and then back by the heart. It will also function with your immune response with the lymphocytes. So our lymphatic capillaries and vessels are more similar to veins than arteries. They have very little muscle in the walls, and they're a drainage system only, so they're not going to be good at pushing fluid through. The lymph nodes are filtering stations along the way for the lymphatic fluid. It does not drain symmetrically. The thoracic duct is going to drain the left arm, the legs, and most of the torso, where the right lymphatic duct is going to drain the head and the right arm. Just like with the veins, you're going to rely on the skeletal muscle pump and respiratory pump. Skeletal muscle pump is going to use those muscle contractions to push the fluid through. respiratory pump is going to rely on the expansion of the lungs through the respiratory system to move fluid through the vessels. So if a person is having problems with blood flow in an area, if they've got edema, they're probably having some issues with the blood pooling in the veins as well. If there's varicosities, there's more likely to be edema, just because the method of transporting both the venous fluid and the lymphatic fluid is so similar. So when we look at lymphatic tissues and organs, we have primary and secondary. The primary is going to be the red marrow and thymus. They're primary because they produce lymphocytes. The secondary is going to be your lymph nodes, spleen, and lymphatic nodules. They are going to provide a location for the lymphocytes to function. The thymus is going to produce your T cells. T cells is short for thymocytes. It's located on the anterior heart. For the most part, this pretty much degrades by adolescence. Your lymph nodes are small filtering stations. There's many locations of these throughout the body. So you generally will have them along your neck and jaw area going down towards your torso. You'll have it in the axillary and groin region. And then there's a fair number of them in the abdominal cavity itself. So any place where you're draining body parts in, you tend to get a lot of lymph nodes. The spleen is a large filtering organ that's going to have many lymphocytes. It functions similar to a lymph node, and even looking at the tissue underneath the microscope, it's going to look very similar to a lymph node. Your lymphatic nodules are large lymph nodes. They're commonly called the tonsils. There's 3 sets of them in the mouth and throat area. Your pharyngeal tonsils or adenoids are in the back of the throat. The palatine tonsils are on the roof of the mouth, and the lingual tonsils are below the tongue. So for people who grew up in the 70s and 80s, it was actually fairly common that people would get their tonsils removed. They do it a lot less frequently now. We've kind of realized that these were a pretty good filter to leave in place. And so it's just not as commonly removed. If they are occluding the airway, chronically infected, then they will still on occasion remove them, but not nearly as frequently as they used to. So when we look at our defenses, we can categorize them as innate or acquired. Innate are immune responses you're born with. A lot of times it's just things about being human that are incompatible for the invader. Acquired defenses are immune responses you acquire with exposure. You've been exposed to something, now your immune system has learned how to react to it. Specific and general immunity. Specific immunity is going to be an immune response to a specific organism or invader or a protein. General immunity are going to be immune responses that are not specific to anything in particular. It's just the body responding to anything that it sees as foreign. So in general, specific immune responses are going to be much stronger. Your first line of defense is a set of physical and chemical barriers to the outside world. The goal of this is to prevent the invader from getting inside the body. First, we have the epidermis. Your skin, it's a physical barrier to a lot of microbes entering. It's good. It's not perfect. Microscopic damage to it can allow microbes to get through, and then there are microbes that actually can penetrate through the skin. Your mucous membranes and their secretions, they're going to provide a sticky, slimy barrier to help trap microbes that may come into the body. The cilia, particularly in the trachea, can help move mucus and particles in the body. This can help move them up and away from the airway. Lacrimal apparatus, so your eyes would produce their tears and secretions. This is to mechanically flush out the eyes. It also has a little bit of the enzyme lysozyme in there. Your gastric juices. So sometimes when some of those invaders want to make their way down into their lungs and they come back up through that ciliary conveyor belt, they end up getting swallowed and going down into the gastrointestinal system. Here it's very acidic, and that will kill a lot of invaders. It's not going to kill everything. If it did, we wouldn't have to worry about food poisoning. Your sweat, it will contain lysozyme. It's on the surface of the skin to make it a less favorable environment. Your skin is not going to be sterile, but what it will do is help to keep the microbial growth under control. So the second line of defense is going to come into play when the microbes have made it past that first line of defense. So we can have inflammation. Inflammation is noted by having swelling, heat, redness, altered movement, and pain. Basically, the body is bringing chemicals into the area to try and control the problem. With a fever, the body raises its temperature to make it a less favorable environment for the bacteria or microbes. It's usually not going to be high enough that it will kill the organisms. If you're going to kill their proteins, you're also probably going to destroy some of your own. But it can get warm enough to slow their growth and allow your other immune cells to get a handle on the infection. Interferon, it's a chemical that will enhance the immune response for intracellular invaders that get inside the cells. But this chemical will also make you feel sick. So while no one really enjoys feeling sick, this can actually be a good indication that your body is working at eliminating the invader. Complement is a chemical released to enhance the immune response as well, but this is going to go after extracellular invaders, and it does not make you feel sick. Natural killer cells, they'll attack anything that's foreign in the body, as well as cancer cells. Macrophages, these are a scavenger that's going to phagocytize microbes, debris, and immune complexes. This is basically going to clean up the damage, clean up the pus after infections. Phagocytes are going to be cells that engulf microbes. Macrophages are one type of phagocyte. They eat up the debris by doing phagocytosis. So when we look at immune responses, specific immune responses, they're going to fall into two categories. The CMI, or cell-mediated immunity. Here, the microbe is inside the cell. This would include viruses. It will also include other things that sometimes will get inside the cell. Here, you cannot remove that organism from the cell, so you have to kill the entire cell. With antibody-mediated or plasma-mediated, it's an extracellular invader. So these would be things that are bacterial, parasitic, or fungal. If they get inside the cell, it's a cell-mediated response. But if they're not, we use the antibody-mediated. One of the important things with our immune system is that we have self-tolerance. We need to recognize our self-proteins so that we avoid killing our own cells. We use that MHC, the major histocompatibility complex, to identify this. Your immunoglobulins are the immune proteins. Antigen-presenting cells are important. When there is an invader, not all invaders are able to process that invader and call for help. Sometimes that cell can't do that, and you have to wait till it does encounter an antigen-presenting cell. What this will do is take part of the protein and display it on the outside of their cell to the other immune cells to get an immune reaction to occur. So when we look at the cell-mediated immune response, once you have presented that antigen out there, the other thing that your immune system is going to need is a co-stimulator. It's a second signal to confirm that it's okay to attack that invader. Your body really does not want to attack itself. So your cytotoxic T cells are only going to be activated if a receptor binds both the antigen and gets a second stimulus to confirm that it's okay to do the attack. A common co-stimulator is IL-2, but there are several others out there. The T helper cell is important here. It releases the IL-2. So it can co-stimulate the B cells for antibody production. It can attract phagocytes to the area and then create more plasma cells. The cytotoxic T cell, this is going to release enzymes to lyse the cell. It's also going to release gamma interferon to help activate the macrophages. So with the cytotoxic T cell, when you have this viral infected cell, it's recognized by the antigen. The cytotoxic T cell is going to kill it. And then gamma interferon will help to activate the macrophages to come clean things up. When you have an antibody-mediated response, here when you have the invader, the plasma cells or B cells will recognize that there's an invader, and they're produced to secrete a particular antibody in response to that infection. So the antibody that they produce will only work towards that one invader. They're also going to require a co-stimulator. And then after the infection, you can have memory B cells. So most of your plasma cells will be recycled, but you will retain some with memory so that if you have any future attacks that involve that same antigen, you'll be able to respond quicker and more aggressively the next time. So this is just a few of the players in the immune system. It is a much more complicated system than what we have time to go into in this class. But this will at least give you an idea of how some of the things are going to interact with each other, the need for the co-stimulator, the fact that we've got the antibody-mediated and cell-mediated immunity. So with this, you're going to neutralize the antigen. The antibody can help to neutralize the antigen. It can help to immobilize bacteria. It's not necessarily going to kill it, but what it can do is stop it from doing its normal activity. You can have antigen agglutination. So with these antibodies on there, it's basically like handcuffing it and taking it out of commission. Complement can be activated, and that will help to enhance phagocytosis. So those agglutinated antigens can be phagocytized and eaten up. So for antibodies, we have different categories. So your IgG, this is present in the lymph and the intestines. It's able to cross the placenta. This is due to its small size. It's active against bacteria and viruses. By being able to cross the placenta, this will give some protection to a newborn baby. With IgA, that's sometimes referred to as secretory IgA, it's present in sweat, tears, saliva, mucus, breast milk, gastrointestinal secretions. By being in breast milk, it can help provide some additional protection to a baby who is breastfed. A baby that is formula fed is not going to get the IgA. It's not the same thing as colostrum. The only way to get that is from the breast milk. So it will act on bacteria and viruses. Levels of this will decrease during stress. So during really stressful times, your mucous membranes will be less protected, and it makes you more susceptible to getting sick. IgM is present in the lymph. This is the first antibody secreted after initial exposure. It's actually a pentamer, so there are 5 of them together. So it's going to have a lot more spots on it for binding to the invader, making it a good option to have as your first one released. It's just more likely to actually stick to the invader with more sticky spots. IgD, this is the least understood of the antibodies. It's present in the blood and lymph and involved with B cell activation. IgE, this one is involved with allergy and hypersensitivity reactions as well as parasitic worm infections. It will react with the mast cells and the basophils. So this concludes our lecture on the circulatory system.